hi, very happy good evening. So this is Krishna Ma'am and we're talking about the story of life on earth, and that's biology. So thank you so much for joining me on this evening. So here we are in our final revision series. So we are starting with the next chapter in biology, that is biological classification. So as you all know, we are doing the one shot series type of revision here. So one day, one chapter. So I'm telling you all the important points. I'm trying to explain you all the important concepts, and I'm also highlighting the topics that you have to revise at the last moment, right? So this is how our revision plan is going, and on Saturday we're going to have a menti quiz of all the chapters we have covered so far. So it will start with living world, biological classification, plant kingdom and animal kingdom. So all the 4 chapters you will have it on Saturday as menti quiz. And for a change, the menti quiz will be in the morning at 11:30 or 12:30, but stay tuned to know the timing. Alright, yes. So today we begin with biological classification. Hi Orchid, a very happy good evening. So thank you so much for joining my lecture this evening. So I hope you will like, share and subscribe. Right, so let's go again. This is going to take us only half an hour because I'm not going to explain all the concepts. So to the points, I have everything summarized in my slides and I'm just going to show you. And if you find my slides very helpful, and if you feel you want to have my slides for your final round of revision, yes, that is possible. So all you have to do is you have to download eCareerPoint app. So log in with an email ID or any Google ID that you already have. So go in. So when you open the eCareerPoint app, so in the downward panel you have a particular panel called as the free stuff. So click onto the free stuff, and there please choose my YouTube channel, that is 𝐒𝐴REERPoint-English medium channel. So please select the series. So we are in the final revision series. So once you select the final revision series, you have all the PPTs in the form of PDF. So you do have access to get these study materials. All right, so that is the undoubted advantage that you have with the YouTube class here at eCareerPoint, right? Hi Ronak, hello, namaste. Yes, thank you so much for joining my lecture today. So please do like, share and subscribe. So without wasting any minute or without delaying any further, we begin with biological classification. All right, so here we go. So we are going to highlight about all these topics. So we are going to see how many kingdom of classification was there. So who did it, why was it accepted, or why was it rejected? So that will be the topic of the hour. So then the first kingdom is kingdom Monida, then it is Protista, then you have fungi, plantae, animalia. So then we'll be talking about viruses, viroids and lichens. Okay, so we begin with the kingdom of classification. So it all began with your 2 kingdom. Yes, the most accepted one is your 5 kingdom, that was given by your R.H. Whittaker. Yes, Chauhan, you're right. But how did it all begin? So it started with your 2 kingdom classification, which was proposed by Linnaeus. The 2 kingdoms were plantae and animalia. On what basis did he classify it? Whether it had cell wall or not. Of course, plant cells had cell wall and animal cells did not have, so it was an easy basis of classification for him. But it was not truly accepted because we were not able to distinguish between unicellular, multicellular. We were not able to distinguish between prokaryotes and eukaryotes. Thus, this two system of classification was rejected. So then it went on to 3 kingdom, which was given by Ernst Haeckel. So here the 3rd kingdom was Protista. So plants and animalia remained, and he gave a new term, that is Protista. So included all the organisms which were not without tissues, meaning only cell level organizations were put here. Okay, but here again we had a drawback or a limitation, that we were not able to distinguish between prokaryotes and eukaryotes, right? Ma'am, pehle RBC and non-RBC pe classify hi nahi. No, they didn't classify based on RBC and non-RBC. No, as far as I know, it was not really accepted. So there was no classification like that. Okay, Chauhan? Right, so this was also rejected. So then we came to the 4 kingdoms. So this was proposed by Copeland. So the 4th kingdom here was Monida. So plantae and animalia remains. Protista came in the 3rd kingdom, and then you have the Protista and the Monidans, right? See, pehle RBC and non-RBC pe. No, that was not a considered mode of classification, so it was ultimately rejected, right? So we do not consider that. So we make sure you read 2 kingdom, 3 kingdom, because you should know why they were rejected. That is the reason we say it here. So based on RBC and RBC, so RBC is cell level classification, right? So that was not actually counted. Okay, Chauhan? Yes? Right. So here the fourth one was Munira. So Munira included all the prokaryotes and your Protista included your algae and fungi. But still people were not able to agree with fungi being classified under Protista. So it was like a heavy burden, so they couldn't tolerate it. So hence this was ruled out. So it was done by Aristotle. So that is the earliest basis of classification. That is all you remember. You do not go further into the details. Okay, Chauhan? Yes? Alright, so then came the 5 kingdom classification. Yes, it was only for animals. Yes, you're right, Nidhi. So hello, thank you so much for joining my session. So please do like, share and subscribe. So then came the 5 kingdom classification, which was proposed by 𝐕𝐵𝐻𝐴. So the 5th kingdom here was fungi. So fungi represents your true mycota and hence it was widely accepted. So even today this entire chapter of biological classification is based on your 5 kingdom. So Munira, Protista, fungi, plantae and animalia. Okay? Yes? But first, not arguing with the teacher. Yes, you're not arguing with me. There's no argument. It's a discussion. You're open to discussion, right? So neither you nor me, both of us are wrong. All of us are learning, right? And it's also a point that I don't know everything in biology. Even I am learning and evolving with you guys, right? So there's a possibility that I might say something wrong because teaching 22 chapters at the same time can be a bit tricky. And I might go wrong. I might have a misconception. That's completely fine. I'm ready to learn. I hope you're also ready to learn. Yes? Chalo. So this is widely accepted. So once it is widely accepted, you should stop. But no, there's another fellow. So that fellow is Carl Woese. He said, no, there's not 5 kingdom, there's 6 kingdom. So here comes the twist to the story. So he said, under Munira, you have 2 groups. One is Archaebacteria, one is Eubacteria. He said the Archaebacteria is a separate kingdom, Eubacteria is a separate kingdom, right? So Archaebacteria, Eubacteria, Protista, fungi, plantae, animalia. So he said totally there are 6 kingdoms, okay? Because, and what basis he classified was, he understood that Archaebacteria lacks peptidoglycan. So Archaebacteria actually are the oldest, the primitive beings. They did not have this peptidoglycan layer. So on that basis, he said there should be Archaebacteria and there should be Eubacteria, right? But he himself did some studies on the 16S RNA of the Munirans and came to a conclusion that there are only 3 domains. Archaebacteria, Eubacteria and Eukarya. They were the only 3 domains, he said, and he did his study on the 16S rRNA. So why was it the study done only on the specific 16S rRNA? So this can be your question of interest because 16S rRNA is not prone to quick evolution. So it is not susceptible to evolution, so it remains the same for a long period of time. So that is the reason he did his research on that 16S rRNA, okay? Yes? No, not like that. So teachers, no, it's not like that. Teachers can be wrong. So sometimes by mistake, I wanted to say something else in my mind, but something else I speak out. So there are possibilities, right? So even we are human beings. There's nothing that I can never be wrong. No, but even if I'm wrong, I'm willing to change. Archaebacteria have cellulosic cell wall. Yes, absolutely right. So I hope this tabular column is clear to you now. Yes? Okay, so he is our Mr. Whittaker, R.H. Robert Harding Whittaker, who gave us the 5 kingdom, right, which we are widely accepting today. On what basis did he classify those 5 kingdoms is very, very important. Based on the 5 points, he classified the 5 kingdoms. The first point being the cell structure. We start with the cell. So how is the cell developed? What is the type of cell? Whether it's a eukaryotic cell, whether it's a prokaryotic cell or a eukaryotic cell. Because we do have a distinct difference between prokaryote and a eukaryote, right? So in a prokaryote, you do not have a well-defined nucleus. Whereas in a eukaryote, you have a nuclear envelope and all the membrane-bound organelles. So based on the type of cell. Next comes the cell wall, whether they have the cell wall or not, on what basis the cell wall is made, whether it is made up of chitin, whether it's made up of cellulose, all these matters. So that is the first point. The second point is thallus organization. So cell later forms the body, right? So thallus is the body organization, whether it is complex or it is simple, whether it is unicellular or multicellular. The first point is cell type, the second one is thallus, right? The third one is nutrition, whether that particular organism is autotrophic or heterotrophic. Sorry, if it's autotrophic, it can make its own food. If it's heterotrophic, it depends on the producers for food, right? And the fourth thing is reproduction. How do they reproduce? Sexual method or asexual method, right? The last one is phylogeny, the evolutionary relationship. So what is their evolutionary relationship? For example, we came from monkeys and apes. They are our recent ancestors, right? So that should also be known. So today, if you want to check something as a drug, so we do it the recent before human trials, we do it in monkeys, right? Yeah. So these are the 5 points on which your 𝐻𝐴 classified these 5 kingdoms, and these points are absolutely important, okay? So talking about kingdom Monera, so he classified based on the 5 points. So I have the 5 points listed here. So that is what we are going to see. So cell type, so all the Monerans are prokaryotes. The cell wall is non-cellulosic, right? So they are made up of your polysaccharides and your amino acids. So here you said archaebacteria have cellulosic cell wall. There can be exceptions, but to a large extent they are made up of polysaccharides and amino acids, okay, right? So nuclear membrane is absent because they are prokaryotes. Their body organization, the thallus formation is cellular. The membrane bound organelles are absent. Once again, it's prokaryotes. Nutrition, they are autotrophic. Some bacteria are capable, your cyanobacteria, they are capable of preparing their own food. So they are autotrophs. So some are heterotrophs, and under heterotrophs they can be saprotrophic. They can feed on dead organisms. They can be parasite, like bacteria causes infection in our body, right? So examples, eubacteria, actinomycetes, blue-green algae, this is your cyanobacteria, archaebacteria, rickettsae and mycoplasma. These are the different examples. So what is so unique about archaebacteria is that they are living fossils. They are the primitive beings. So since the time life originated on earth, it is this archaebacteria that lived, right? So they are the primitive beings. What is their cell design? They have polysaccharide. So in my slide, PS is polysaccharide, PP is polypeptide, and they have branched lipids. So when I say archaebacteria, one more thing should strike your mind. So that is, they live in the extremes of conditions. So why do they live in extreme conditions? Is exactly, okay, because they have this branched lipid. They were able to survive it because of the branched lipids, okay, right? So the types of archaebacteria, methanogens, which can produce your methane, like the one that you read in class 12, the sewage treatment, right? Then halophiles, which can tolerate a high amount of saline condition. Thermoacidophiles, this is one term. Thermo is temperature, acido is acidic pH, so they can survive in both, like your Thermus aquaticus, right? So the different types of archaebacteria and their example is given below. The methanogens, they produce methane. The examples include your methanobacterium, methanococcus, rumenococcus, right? Sorry, methanobacterium is one name. Methanococcus, rumenococcus are two different examples, okay? Halophiles, they have a purple membrane. So when you see towards the coast of some places, the tides, the waves are purple in color because of the purple membrane of these halophiles. Halobacterium, halococcus is one name. Okay, so here I have 3 different members. Here I have only one bacteria. Halobacterium, halococcus is one name. Thermoacidophiles, they are basically aerobes. So this question has been repeated. So thermoacidophiles are aerobes, and anaerobes are both. So they are strictly aerobes. The temperature, they can tolerate high temperature of 80 degree and they can tolerate an acidic pH of 2. So the example, Thermus aquaticus. So sorry, this is S, Thermus aquaticus. Sulfobolus, thermoplasma, these are the examples. I want you to remember the examples. Okay, eubacteria, how is the eubacteria designed? The eubacteria does not have a cell wall outside. So before the cell wall, it has what is called as a glycocalyx layer. Then it has a cell wall. Then it has a plasma membrane. Okay, so this glycocalyx can be very tightly packed. It is a capsule. It can be like a sheet that is called as a slime mold. It can take into various forms, right? So now, depending on the cell wall, you can have gram-positive and gram-negative bacteria, right? So gram-positive bacteria, as you can see, they have a big peptidoglycan layer. Then you have the phospholipid bilayer. But in a gram-negative bacteria, the peptidoglycan layer is sandwiched between the two phospholipid bilayers, right? Yes? Okay, so that is the main difference. So gram-positive bacteria can retain the stain. Suppose I apply mehendi, right? So after an hour or two, I wash my hands. So the orange color stain of the mehendi will stick to my hands, right? That is your gram-positive bacteria. Imagine I apply mehendi, after 1 hour, my hands become like this normal. That is my gram-negative bacteria. So gram-negative bacteria is not able to retain the stain. So in case of bacteria, I'm not going to apply mehendi. I'm going to apply a dye, a colored dye. But that colored dye will not stay inside the gram-negative bacteria because of the thin peptidoglycan layer. So it has an affinity to come out again. So your gram-positive bacteria will retain the stain and a gram-negative bacteria will not retain the stain, right? That is the main difference. Yes, W. Roy, a very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. So W, I saw your question in Telegram. So you have a doubt in recombination. So your longest difference will be the highest recombinant one. Shayad your BC has a recombination of 28%, so they are in the corners, right? So it is understood if B and C are in the corners, in between it is A, so hence your last option B, A, C is the right answer. Okay, W? Yes? If you still don't understand, ping me again, I will explain it there, okay? Yeah, coming back here, so you can read, so this has low lipid content because it has a thick peptidoglycan layer. So this has high lipid content because it is sandwiched between the phospholipid bilayer. So your gram-positive are not pathogenic, whereas gram-negative are pathogenic. So you'll remember the examples of both, right? Yes. So based on motility, so I hope you know flagella helps in motility. Based on where your flagella is present, there are 3 types. Okay, W, please remind me at the end. I will explain. Okay. Right. So if it's A trichus, it has no flagella. If it is mono, it has one flagellum at one end. If it is cephalotrichus, it is present at one end. A bunch is present at one end. Okay. So this is your cephalotrichus. Amphitrichus, it is present on both the ends, and peritrichus throughout the entire perimeter of the organism, and you can remember the organisms. So this is actually not for NEET, but you can mark it if you're going for any other exam where you have much depth of bio. You can make sure you go through it. Okay. Yes. What is stain? A stain is a dye. For example, under the microscope, if I have to see a cell, I will not be able to distinguish between which is a mitochondria, which is a nuclear membrane, which is your cytoplasm. So I color it with a stain. So I take a glass slide, I put my cell, right? I cannot see it. So on top of that, I put a drop of dye, any colored dye. For example, let's say methylene blue is a dye, I put it, okay? So now I let it dry so that the dye gets absorbed, and when I view it under the microscope, so methylene blue, say for example, it is blue color. So under the microscope, wherever it is blue color, that is the part I want to see. That is a stain, that is a dye. Okay, Gautam? Yes, fine. So next we are going to talk about cyanobacteria. So they are blue-green algae, right? So they are gram-negative. They do oxygenic photosynthesis, meaning they release oxygen. So they have granules and they have a structure called heterocyst, which helps in fixing nitrogen. So this last point is very important, this heterocyst part. So make sure you remember it. Fine. So mycoplasma, they are the smallest. They have no cell wall. So all monirans have cell wall except your mycoplasma. So since there is no cell wall, there is no rigidity. So it can make itself into any number of shape and form, and that is why we call it as pleomorphism. Pleo is many external ways. Okay, they are again gram negative and this is how they reproduce. Then you have actinomycetes. The actinomycetes are today known as mycobacterium. So there was a confusion whether to put actinomycetes in fungi or in bacteria because it had a fungal-like structure, but it worked similar to a bacterium. So after deliberate argument and discussion, they have placed it under the kingdom Monera. Alright, fine. So coming to the next one, kingdom Protista. So we are all single-celled eukaryotes are protistans. So protistans are mostly aquatic, they live in water. So protistans can be classified based on their mode of nutrition. If they are autotrophic, you have dinoflagellates, diatoms, euglenoids. If they are slime molds, if they are and feeding on dead organic matters, they are slime molds. If they are protozoans, that is, if they are parasites, they can be your protozoans, right? So we look into it. So how do they reproduce? They reproduce by fusion. So gamete is fusion. Fusion of male and female gametes is gamete, okay? So syngamy, when both the gametes come together, that is normal fusion, okay? Yes? This is NCERT line. Which one is NCERT line? This one. Syngamy. Yes, this is the concept that is mentioned in your NCERT. But there can be different types of fusion. For example, if you look isogamy, both the male and female gamete are of the same size and they are genetically similar. So it is isogamy. Anisogamy, one is big, one is small. So it is anisogamy. When you see oogamy, one is non-motile, one is motile, and the size is also different. This is oogamy, right? So life cycle of a protist has two types of life cycle. One is zygotic meiosis, one is gametic meiosis. So right after zygote formation, if there is meiosis, it is zygotic meiosis. So imagine if the protist is haploid. If the adult protist itself is haploid, don't confuse, so it will form a haploid one. So right after zygote, you are having meiosis, so it is zygotic meiosis. What is gametic meiosis? So if at all the protist itself is diploid, right? So now if I have to produce the gamete, it will undergo meiosis here. So if the meiosis happens to form the gametes, it is gametic meiosis, and the examples are given in your slide, which you will go through. Fine? Yes? So who are the members of your protist? Diatoms, dinoflagellates, euglenoids, slime molds, then protozoans. So when you talk about diatoms, all that you have to remember is your soap box. Okay, so how is your soap box? You have one base box and a lid. That is how it looks like. The walls of these diatoms are very, very strong because they are made of silica. So they are very, very tough to destroy. The examples of diatoms are Navicula, Cyclotella, Pinularia. So remember physics and chemistry. Pinularia, Navicula, Cyclotella. So here the cell wall is made up of silica, right? So dinoflagellates, the cell wall is very stiff. They are not made up of silica, they are made up of normal cellulose, but it is very thick. Diatoms are also called as chrysophytes, okay? Or you can say chrysophytes come under diatom, either way is right, okay? So this is very stiff, but one important criteria is bioluminescence. They can light themselves, right? So they can cause red tides, example Gonelex. Euglenoids have no cell wall. The first one had silicaceous cell wall. This had a very stiff cellulosic cell wall. This does not have a cell wall. But instead to compensate it, it has a pellicle protein layer. When sunlight is there, it will perform photosynthesis. When there is no sunlight, it will become heterotrophic, right? So example is your euglena. The last one is slime molds. They are saprophytes. They feed on dead organic matter, right? And the example is your Physarum. So Physarum is a type of your slime mold, right? So coming to protozoans, they can live as predators, parasites, etc. There are 4 types. The first is amoeboid protozoan. So they live in fresh water. So they have the false feet. Example is your amoeba. Flagellated protozoans having flagella. Example, your trypanosoma, which causes your sleeping sickness. Ciliated protozoans, which has cilia structures around the body. Example, paramecium. The last one is sporozoans. In human health and disease, you read about the life cycle of plasmodium, right? So they have a particular infectious stage called as the sporozoid stage. So that is your sporozoan state, right? You're able to recollect everything? Okay, so coming to the last part of the chapter, the kingdom fungi. So fungi is mostly found in humus-rich soil. They live parasitically, meaning they can cause diseases. The black fungus, the white fungus. So towards the end of the pandemic days, so we had to fight with the fungus also, right? Yeah, so fungus also grows on the bread mold. So it is parasite. So chloroplast is absent. So fungi are basically heterotroph. So there is no fungi which is autotrophic. So fungi are heterotroph, right? They can obtain food in various ways. They can act as parasites. They can act as saprophytes. They can feed on dead beings, right? So they can also live as symbionts. Lichens, your algae and fungi have a mutualistic understanding, right? So all this. So what are the different classes of fungi? Okay, so sexual reproduction of fungi. So if a fungi has to reproduce, so imagine I have one fungus here and I have another fungi here. Fungi are multicellular, right? So now if these two has to fuse, first the plasma membrane will fuse. That is plasmogamy, fusion of plasma membrane. So now there is two nucleus. Now the nucleus should fuse. This is karyogamy. Karyokinesis is nuclear division in your cell cycle and division. So karyogamy, fusion of the nucleus, right? Okay. So once this is formed, this will undergo meiosis to form your haploid individual. So manlo, if this is haploid, this is diploid. So this becomes haploid once again. But it's not necessarily that both the nucleus fuse together immediately. It can enter a intermediate phase, that is your dikaryon phase. Fine? Yeast is a only unicellular fungi. Yes, you're absolutely right. So you have four members or four classes of fungi: Phycomycetes, Ascomycetes, Basidomycetes and Deuteromycetes, right? So Phycomycetes, they live in aquatic habitat, decaying moist soil, all that is fine. So they are aseptate. So fungi has a structure called as hyphae. Network of hyphae is mycelium. So if my mycelium is like this, it is divided into compartments. If it's divided into compartments, it is septate. If it has no compartments, it is aseptate. What is coenocyte? It can have many number of nucleus. That is coenocytic condition. Okay. So zoospores here are motile, aplanospores are non-motile. Okay. Example, Rhizobus, albuco and mucor. So here you will remember the examples as RAM. Fine. The next one is Ascomycetes. They are sac fungi. There is a bag-like structure. They are saprophytes, decomposers, etc. Coprophilous is they grow on cow dung. Their mycelium is branched and septate. If you see, the only fungi which is aseptate is your Phycomycetes. So if this question comes, it is very easy to answer, right? Asexual spores have conida because fungi can reproduce in all the three ways: vegetative reproduction, asexual reproduction and sexual reproduction. So the asexual spore is conida and conidiospores. Sexual spores are ascospores. So the example here is Aspergillus, claviceps and Neurospora. So here it stands for can. But there are two more Ascomycetes which are edible. So they are morels and truffles. So these two are edible. You can consume them. So remember trophies and munches, so that is edible, right? So Basidomycetes, they are mostly parasites. Their mycelium is branched and septate. They have basidospores arranged in structures called as basidocarps. The example is Agaricus, Ustilago and Pucina. So Pucina causes a disease, Ustilago causes disease and Agaricus is a mushroom. So I remember it as all people are useless. True, no? Yes? At one point or the other, we are causing harm to the earth. Right. The last one is deuteromycete. It is imperfect because it cannot reproduce sexually. Right. So, example, Alternaria, Coleotrichum and Trichoderma. So, here this I remember it as cat. So, read it thrice, you will remember it automatically. That is the secret, okay. So, I have been teaching this over the years, so I remember it by heart. Fine. So, fungi is done. So, the last topic is viruses, viroids, prions and lichens. So, you know virus is non-living. So, unless and until it is an inert organism. So, unless and until it enters a living human being, it cannot function because it needs a host to replicate itself, right. So, a virus can have DNA virus, the genetic material can be DNA or RNA. At the same time, it will not have both DNA and RNA as a genetic material. That is not possible. But for your kind information, yes, we have DNA viruses. Okay? Yes? Right. So, viruses can infect plants, all that you know. So, there can be a protein coat surrounding the genetic material. So, if this is the genetic material of a virus and if I have a covering like this is a protein covering. So, this covering is a capsid. So, this capsid can have many points or many subunits, that is capsomeres, right? Yes. So, example of virus, your mumps, your smallpox, your coronavirus, etc. So, now what is a viroid? So, this protein coat is absent in a viroid. So, it only has a genetic material. So, it is comparatively less in weight compared to a virus, right? So, it has only RNA, no protein coat. What are prions? Abnormally folded proteins which can cause infection. Example of the disease which is caused by prion is bovine spongiform encephalopathy, right? So, who are lichens? The symbiotic association. Bacteriophage has double-stranded DNA. It can even have RNA. Bacteriophage is a virus infecting RNA. Sorry, is a virus infecting bacteria. So, it can be a DNA virus, it can be an RNA virus. It can be a single-stranded RNA, it can be a double-stranded RNA, or it can be a double-stranded DNA. Okay, Orchid? Yes? Yeah. So, algal component is called as phycobionts and the fungal component is called as the mycobiont, right? So, with this we come to the end of biological classification. Was this quick revision helpful to all of you? Yes. So, before we forget, let me finish off W's question. So, W had a question from Principles of Inheritance and Variation which he pinged me in Telegram. So, the question was about mapping frequency, right? So, the question said B and C had a frequency of 28%, right? And if I remember, AB had a frequency of 8%. And what is the remaining? AC had a frequency of 20%, right? So, the maximum is 28. So, this is B and C. So, AB, so A can be here. So, this is 8% and this can be 20%. So, the order of the gene is BAC. Okay? Yes? Ma'am, Hindi mein bhi padhaya karo. Okay. So, but this is an English medium channel. What do I do? So, you have a Hindi channel separately. So, this is for all the students who cannot understand teaching in Hindi. So, that is the main reason this channel was started. If you want, I can slow down, and if you do not understand, I can help you with Hindi. But you have to try to learn it English, because later in your medicine or in your IIT, you have to read it in English, right? Hindi medium ke bachche NEET kaise padhenge? Alright, so I understand you're from completely Hindi medium background, so you do not understand the English I speak. But my advice is koshish to karo. Thoda try to understand, so that in future it will not be tough. Ultimately, there is no NEET or there is no JEE which is completely in Hindi, right? Thoda koshish karo. There is a Hindi medium channel where they teach in mixed language. But still, if you can't understand, please ping me in Telegram. Whatever I'm teaching you, I'll try to translate in Hindi. Alright? Yes? Don't worry, it's not a criteria to feel low. It's an opportunity to grow. Alright? Yes? Yes. So, W, I hope you have understood the question. For all those, so biological classification is done. Yes, Orchid. So Orchid says yes and she's understood the quick revision. So it's my humble request, so please watch this video on heat box. It is very interesting. So it is uploaded in this channel. So if you type heat box by Krishna Ma'am, you get this thumbnail. So please watch, it is very interesting. So towards the second half, I would have spoken about the pox. What is that pox? So recently World Health Organization has declared it as the pandemic, right? So please listen to it. It's just a 7 minutes video, so I don't think it is a very long video to watch. You can still try your best, okay? And for all those who do not know what is the quota coaching, and if you've never been to quota, there's a beautiful opportunity to be in quota with me, with us. So we have our motivational guru, Mr. Pramod Maheshwari, who's the director and CMD of 𝐒areerPoint. So he is going to interact with students talking about the 6.5 mantras for success. So it's the best way to know what does the quota coaching look like. So people have a great hype about quota, right? So quota is a hub for coaching. So what it is, so how do we actually believe what is happening? So please join us offline. It's not any Zoom session or anything. So we are organizing this in the CP Tower auditorium. So you're most welcome. The event is on 8th of June. Even now, if you book your tickets, you can be here. So please, you have a lot of promotions going on this, so you have to fill your form. So beneath my video description you have a registration form, so please fill it and please be there. So it's a chance that we can also get to meet. So thank you once again for this promotion. So I hope I'll see you here. So if you genuinely liked my videos and my way of teaching, so please don't forget to like, share and subscribe. So I'll meet you tomorrow at 5:30 with the final revision of Plant Kingdom. So until then, it's your time to do self-study. This is me signing off. So take care and bye. Thank you guys.